Thank you guys for being here. After our lunch break, we have our keynote speaker, Dr. Martin Chalfie, and it gives me great pleasure to introduce him. He's a university professor at Columbia and shared the 2008 Nobel Prize in Chemistry for his introduction of GFP, Green Fluorescent Protein, as a biological marker. He got his BA and PhD degrees from Harvard and did postdoctoral research with Sydney Brenner at the MRC Laboratory of Molecular Biology. His research uses genetics to address how different types of nerve cells acquire and maintain their unique characteristics and how sensory cells respond to mechanical signals. He's currently the chair of the Committee on Human Rights of the National Academies of Science, Engineering, and Medicine, and in January 2021 becomes the president-elect of the American Society of Cell Biology. So thank you so much for being here, and I'll turn it over to you.
Thank you, Arya. It's a real pleasure to be asked to speak. I have to admit, I have not really spoken to journalists before, science journalists, about science journalism. And so I've been thinking about a number of things that I might want to talk about. But all of them revolve around writing and my experience in writing. So let me start the slides. I'll share the screen and start the slides. And we can begin. So as my title says, I feel that writing is not really the summation of some research project, but really an essential component of research. So I'm really going to tell you about 3 or 4 different things, all of which are somewhat unrelated to each other, but have the common theme of writing. And I've put my email address here if anyone has any questions that I'm not able to deal with during the time that we've been allotted. So I'd like to start, during this pandemic, I've caught up on some of the reading that I have always wanted to do. And one was a book written in 1983 by Daniel Boorstin called The Discoverers, which is a really quite wonderful book. He was the Librarian of Congress. And it really is a wonderful book, but I was particularly struck by the passage that he had on Leonardo da Vinci, one of my heroes in science, and about his work really advancing people's concepts of anatomy. And then after he said all these wonderful things that Da Vinci did, he wrote the following. He said, despite his consummate art, his industry, and his unexcelled powers of observations, Leonardo da Vinci added only to his own knowledge and little or nothing to the anatomical knowledge of his time. Nor were his own observations enriched as they might have been, for the public forum of printed matter has a way of improving the product, and Leonardo's work remained private. This is a very different way of thinking about Leonardo's work as basically the selfish scientist not sharing his knowledge with the world and losing out because of that. And so again, writing is very important. My own history or my path to scientific writing did not start off well. I tried to get into an advanced English class when I was in high school. I think I was in there for about a month or two, and it was deemed by everybody involved that I was somewhat of a failure, and I had to go back to another English class. So I was booted out. And all through my early career as a scientist, there were hints that I needed to have a lot of help in writing. In fact, when I was finishing my PhD thesis with Bob Perlman, the way he informed me that it was time for me to write my thesis was not to just come out and say, you know, you've done enough work, you should write a thesis. He gave me this book, Elements of Style by Strunk and White, told me that I had better read it from cover to cover, basically memorize it, and then write my thesis. So I had the hint that I needed some help. But in fact, all the material that I learned in high school, college, and even through this wonderful book, Elements of Style, were about grammar. I really didn't have a sense about what to do as a writer until around 20 or so years ago, when I found out about a wonderful article that I now give to everybody in my lab and anyone who ever talks to me about science. So I'd like to share it with you guys. It's an article by George Gopin and Judith Swan called The Science of Scientific Writing. And the reference is here in the slide. It's a really wonderful article because they say, yes, you should know grammar and you should try to avoid the passive voice, but you really should be doing something much more in your writing. And that is that to know that if the reader is to grasp what the writer means, the writer must understand what the reader needs. In other words, we have to think about how people read and think about the writing to be able to write clearly. And they present a number of rules, which I had never seen anywhere before, and have changed my writing and the writing that people in my lab do. So one of the rules could be described in the following way. The subject and verb, because everything between them is viewed as parenthetical, should be close together. Well, this is clearly the wrong way. If this is really important, the right way of writing this is the subject and verb should be close together because everything between them is viewed as parenthetical. But it is astonishing how much scientific writing is done in which the subject is separated from the verb by about a mile of words. And so it makes it almost impossible to understand because we don't pay attention to it. The other rule that they say, which is so simple as this one, but a surprise to someone who hasn't thought about writing, such as myself. And that rule is that the subject is old information and the object in the sentence is new information. So for any sentence, A somehow is changing to B, old information to new information. And what's needed is to connect up that sentence with the next one, which would be B or B prime, is somehow changed into a new idea C, C going to D, and D going somehow to E. And when you finish this chain, that's the end of the paragraph. And it gives a very nice definition to how the flow of information should go in a paragraph. And I can tell you, every person I've ever shown these to has said, I've never heard these rules before. So it's been exceptionally useful in being able to convey information. And it's surprising, for example, in this last point, how I will read someone's paper, a draft of a paper, and it will be that one of the connecting sentences is missing. And once it's put in, everything makes sense. Anyway, let me get to my real point here about the importance of writing for scientific research. The usual way people work in a lab is the first thing you do is a series of experiments. And then you might report on these experiments to your advisor maybe once a week and tell him or her what those results are. Maybe periodically, every couple of months or so, one would meet with the whole lab and describe the work in a lab meeting. And this is usually where we're left off in thinking about how we should present stuff. And these meetings with an advisor or mentor or the lab meetings where you present to a larger group of people are very useful because they're great ways to discuss immediate problems and get feedback. The problem, however, is these are usually not opportunities to think about the whole project. You're only talking about the latest problems or successes that you've had since the last time you talked. So there's never a chance to think about this overall. That is, until you write up the results. And once you write up the results, that's really often for people the first opportunity to consider the entire project. I might add parenthetically that one of the consequences of this, of realizing this for my lab, is that I now have people in my lab every year, year and a half, if they haven't written up a project, to actually write up a project so they get this overview of all that they've been working on. In any case, once this is written and the entire project is considered, the entire project may change. And that means that, oops, let me go back to that. That means that in many cases, writing is actually the beginning or maybe the middle of a project, but certainly not the end of it, because lots of things change. And I want to give an example from my lab and some work that we did several years ago. We work on the touch sensing neurons of the nematode Cinerabditis elegans, and here they are shown with GFP to highlight only those cells in the animal. And over the years, we've identified transcription factors that are needed in the animal for the animal to generate these cells. Two genes encoding these transcription factors are UNC86 and MEC3, and together, UNC86 turns on MEC3, then MEC3 and UNC86 turn on all the genes needed for the touch sensing cells, and they also are needed to keep MEC3 up at a high level. And we were pretty happy when we had this. We figured we had all the answers to how the cell was generated and maintained its development. And then a new postdoc in the lab many years ago, Irini Topolito, did more experiments, and she found a third transcription factor, one that was turned on by UNC86 and MEC3, called ALR1. And she did all the standard work to understand this gene. She looked to where it was expressed. Was it expressed in these 6 cells? And the answer was yes. She asked, did this gene on its own activate any genes? And the answer was no, on its own, it did not. But when it was with MEC3 and UNC86, it doubled their activity. So this was a nice piece of work. She also looked to see, because these are touch sensing cells, she also looked to see, were the animals affected in their touch sensitivity? And the answer was yes, but in a very strange way. Normally what we do is we touch the animals 10 times, and we ask how many times out of 10 did they move away from the touch? And in wild type, they almost always move away from the touch. But in the ALR mutants, and each one of the dots represents the response of one animal, in ALR1, some of the animals had no response, and some of them were virtually wild type in their response. There was an incredible amount of variability. And it was this variability that really intrigued us, because we realized after several more experiments that I won't go into now, that ALR1 was serving a very different function. It was not there to turn on genes. It was there to make sure that MEC3 and UNC86 turned on the genes. And this is a fundamental problem in biology. How do you have high fidelity actions taking place? And we realized that ALR1 bound very close to where UNC86 MEC3 bound, and so it enabled them to work at a much greater efficiency, so every cell developed instead of only some of the cells developing. So it wasn't needed for transcription. It ensured that UNC86 and MEC3 acted optimally, and so we refer to this as a really different type of transcription factor called a guarantor. Well, almost all of that work happened after she wrote the paper that we looked at, because once we were able to look at these data, we realized there was something much more interesting that we wanted to talk about and to study. And we eventually found that this guarantor function was an important function, not only to keep MEC3 on, but we found other guarantors later that actually turn on UNC86 and start the whole process. So this idea of guarantors came out of looking at a completed bit of work that then changed the paper enormously. want to switch now to a completely different idea. Arya said that I was privileged to get the Nobel Prize in 2008. The work that we published on that won that prize had some problems with publishing, so I'd like to quickly go over what those problems were. We sent the paper to the journal Science, and the first thing they did was they actually rejected the paper, and they rejected it because the title was something they did not like. And so they rejected it on the basis of the title. The title was Green Fluorescent Protein, a new marker for gene expression. And when I asked them why they rejected the paper because of the title, they said, well, you see, they're a little haughty at Science. They said, you see, everything that we publish is new. And so you cannot use the word new in the title. After a little bit of negotiation, I asked them if I could change the title. And they said yes, and we resubmitted the paper with the following title, which is, you can tell how angry I was. The title is The Aquaria Green Fluorescent Protein Needs No Exogenously Added Component to Produce a Fluorescent Product in Prokaryotic and Eukaryotic Cells. And the paper was accepted, which was quite nice. And then the editor called me up and said, you know, your title is quite long. And I realized that because one, I was angry, and two, it was almost the entire paper. And I said, would you like me to change it? And they said yes. And I said, well, how about this? And I changed to green fluorescent protein as a marker for gene expression. And that's what the official title is. The second problem with publishing has to do with the picture on the cover that you see on the left of this slide. I had taken this picture of a living worm, and I wanted it to be shown on the cover because it would show that GFP could be used in living organisms. And the cover editor called me up, said that she really liked the cover picture and wanted to use it, except they had a problem. And the problem was that it was the wrong color. They apparently never like to have green on the cover of their magazine because it doesn't show up very well. And in fact, this is a enhanced picture of the cover. It does not look very well, but I could not let them change the color of green fluorescent protein. And the final problem was that we were using work by other people that we had given away samples, and they were reporting back to us that GFP was working in other organisms, and we wanted to cite that. And almost everybody gave us permission except one person. Here's her picture and her letter, which you don't have to read because she demanded that I make coffee every Saturday morning for the next 2 months and prepare a French dinner and take out the garbage nightly for a month in order for me to be able to cite her work. This is my wife, Tula Hazelrig, and the work that she did was actually spectacular because she made the first protein fusion with GFP and was able to watch it in developing Drosophila eggs. I want to finish with a plea, a suggestion of things that I hope people will go back to their own individual labs and suggest that they do. As some of you may know, there's been a big push over the last few years to have people in biology do what the people in physics have done for about 27 or 28 years. And that is, when they have a paper completed, to not only submit it to a journal, but to also put it in a preprint archive. And the work in biology has been really spearheaded by these 4 people in an organization called ASAP Bio. And its website is asapbio.org. And what this is to promote is to get people to submit their articles so that basically everyone in the world can see them. To me, the real advantage of putting things in a preprint server is you don't have to be at a rich university, you don't have to be in a rich country, you don't have to be at a university to be able to get access to biological reports. You have to know that it's not peer reviewed, but at least you have access to the data and can think about it. But as much as I'd like to talk about the whole idea about preprints, and the preprint server we use is the one at Cold Spring Harbor BioArchive, I really want to promote an idea that I first heard from this man, Francis Collins, who's the current head of the National Institutes of Health. We were talking at a meeting of ASAP Bio about this, and he told me about a wonderful thing that he does with his lab. And I've been doing it with my lab for the last 2 years. We have not a lot of people in the lab. So there's people sort of balk a bit when I say they have to give another lab meeting. But the way we've arranged things is that people talk about their work 1 week, and we alternate that with a journal club. But the journal club has a very specific rule, and that rule is the paper cannot be published. It must be from bioarchive or even the physics archive. And they therefore are writing about articles that they found that are interesting that have probably only been put on the archive in a month or two. So there's a lot of advantages to this. And I should also say that we have a particular procedure that we go through. Every single person in the lab presents. That means undergraduates, graduate students, postdocs. Everyone gets together for about an hour and a half, and we talk about the paper, and the person in charge of the discussion writes up all the ideas that we have, and we send them to the authors. So for the lab members, they get to read the latest material of interest. They build their skills in reading papers and reviewing things, but at the same time, they realize it's not an empty exercise, that they're actually doing something useful and helpful to others. The authors obviously get additional reviews and feedback. And I should point out that we're not trying to judge whether the paper should be published in a particular journal or not. We're interested in the science, not its worthiness for publication. And the actual goal of all of this, and why I'm promoting it to all of you, is that what we really want to promote is a sense of sharing and building a community, because we're actually all in this together, and being able to share ideas and give feedback to people is exceptionally useful, and it's part of being a good scientific citizen in my point of view. So those are the points I want to make. I want to thank you for listening, and I guess there's a little bit of time we can take some questions. So thank you.
Thank you so much, Dr. Chalfie. That was fantastic. I guess we'll start off in the order that questions are received in. So go ahead and put your questions into the Q&A. I'll read them off, and Dr. Chalfie can stab at them. So let's start with, in a few papers I've read, I've noticed that researchers might administer a drug in an ineffective way in the control group, for instance, orally versus intravenously, in order to yield more significant results in their experimental group. So if we're reading a paper in a field we are not well versed in, how might we go about identifying these types of hacks that researchers might use?
So that's a very interesting question. Usually this is what precisely reviewers pick up. And I think that gaining this skill of being in the journal clubs and having people discuss problems and saying, you know, I noticed that they didn't do this. Part of that is increasing one's awareness of these issues. So in a sense, it's a little bit of practice.
Okay, great. We'll take the next question from Harrison. Are the papers reviewed by the club in manuscript form or following their publication? I think he means your lab meetings.
So in my lab meetings, we go to bioarchive.org. We find a paper that is of interest to us. I don't think this would work on a department-wide basis or a class necessarily, but we're interested in a lot of different subjects in my lab. And so people usually find a paper that's related to it. And that manuscript is on bioarchives and can be downloaded as a PDF with all the images and the supplemental material, everything one needs to read the paper. So no, in fact, it shouldn't be published.
Okay, great. When you write manuscripts, do you keep a specific audience in mind?
Not necessarily. We want to, you want to make as many connections as possible when you write the discussion. You want to make it broad. We're writing to a scientific audience. We're not writing to a lay audience. But for many journals, they do ask for a lay summary, which I think is very useful. So we sort of direct things in many different ways. It depends on the journal and what they're asking for, but there sometimes is a requirement to have a lay description so that a much broader audience can understand and read the paper. We also don't, personally, I don't like writing things that make it so obscure that no one wants to read the paper. I mean, we do want to have some sort of general import of the paper that we're writing.
Okay, great. And this is the final question we have right now. So if you have any more questions, I'm going to ask that you put them in the Q&A now. And here's the question. I definitely believe that scientific miscommunication is perpetuated by how a scientist writes. Like the example you gave with the subject-verb separation, how should we implement this in undergraduate's curriculum or mitigate it?
So I can only give you an example that was actually, so my wife is also a faculty member here at Columbia, and she was in charge 1 year, and then the next year I was in charge and liked her idea so much I did the same thing, is that she was in charge of we have an independent study program, and at the same time we have a course for the people taking independent research on bioethics, other issues, and writing and putting things together. And she spent, and I spent the next year, quite a lot of time with students working on the reports of their independent research projects, which they had to write at the end of the second semester of their independent research. And the way we did it was to have groups of no more than 4 students. Each week, they would write up a section of their report. So for example, they might write the materials and methods, or they might write the introduction, or the abstract, or the results, or the discussion, one of those parts of the paper. And the class met only for an hour, I think, on a Tuesday. By the Saturday before the Tuesday, they had to send the other 3 people in their group what they had written. And then the whole group would look at everyone's paper, and really from the one point of view, they could improve the writing if they wanted, but the really important perspective was, did I understand what you were saying? And that feedback was very, very helpful. In fact, after the students had gone through this, one of the faculty members that did not know what we had been doing suddenly wrote to the whole faculty. He was one of the people grading these things, and he was looking through it, and he wrote to the faculty. He said, "We have just gotten the best students we've ever had in our program." And he didn't realize that what he was doing is he was reading these much more refined papers. And what it says is people can learn. People can make their writing clearer and more understandable to a broader audience. And that's very, very important.
Okay, we have 2 more questions and then we'll wrap it up.
Okay.
We'll start with, do we as scientists have a responsibility to produce good research, or is that on reviewers to decide?
You know, there's a disparagement that's sometimes said about research, that people outside of research say it's only curiosity driven, or it's, it's ivory tower science. And that's really bad-mouthing science, because in fact, if you're going to do some work, you should be doing work that you have a good idea of why you're doing the work. So I'm going to interpret the question of good science as, why are you doing the experiment? If you're doing the experiment just to repeat what 20 other people have done, that doesn't sound to me like good science. But if you have an idea, it's not that it has to be has to be on a disease or it has to be anything else. In fact, most discoveries really come from way outside what anybody else has been thinking. There's a wonderful line by the physicist Enrico Fermi that goes something on the order: if you do an experiment and it confirms your hypothesis, then you've made a measurement. But if it doesn't confirm your hypothesis, then you've made a discovery. And those discoveries come all the time. But I do think that people do have to think about why they are doing an experiment. The answer my advisor told me to is not the right answer to that, but really trying to get at what that question is a very important part of being a scientist. And the last question?
Yes, last question. How can students learn to take up space and push some of the ideas you presented? For instance, persuading lab members to see the value in the time commitment for these sorts of reading and writing exercises.
So I don't think it's going to be that hard. I hope it's not that hard, to tell you the truth. I can tell you that we've been doing this now for about 2 years. There's not a single time that any head of a lab has come back to me and said, Marty, I never want you in your lab to read another one of my papers again. In fact, what they almost all come back and say is, I really like this idea. We're going to institute it in our lab. And so we've gotten a lot of very positive feedback. If you like the idea, I would say just speak up, because it's part of your own training. It's what's going to make you, I hope, a more involved scientist. And I suspect that people will thank you for bringing the idea of how to get a jump on things. I mean, a lot of times published papers have gone through at least a year of review before they're actually published. And so what you're really reading is work that's at least a year old, but that work is only about a week old or 2 if you get it out of the bioarchive, for example.
Okay, great. Thank you so much, Dr. Chalfie. We really appreciate you coming to NUCSJ and speaking with us today.